Hi there.
Welcome to Microbiology Twitter Journal Club, where we nerd out big about all things small.
Oop, oh my goodness. The music on this side. My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin egg with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated to enabling the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but I also worked as a research integrity specialist. I'm currently working as an editor for a scientific journal.
Next week, or sorry, every week we meet to talk about microbiology, and today it's our news week, where we do an overview of papers. Well, I guess it's a special news week.
Yeah.
We're doing an overview of papers we've seen in the news. In this case, the top 10 cited papers from 2020 that are non-COVID-19 related, to try to find an article that we can discuss next week.
And next week will be our deep dive week, where we take that article and we really delve into the depths and look at it figure by figure to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
And as always, you can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments to tweet us using the hashtag MicroTWJC hashtag. Yeah, so this week we're doing something really different because around the world, different countries are starting to celebrate not celebrate, commemorate, because it's not really something we want to celebrate, but the COVID anniversary where we've spent, because so I think it's been a year since we've officially went into the lockdown, and so I felt the best way to kind of commemorate this was to literally not talk about COVID. We've
taken a break.
We've spent an entire year talking about COVID and COVID research. So I think we should focus on the microbiology articles which we might have missed because of COVID.
Yeah, and that was always the intention with our channel, right? Like we started because it was like something interesting, like it was on our minds. We wanted to keep up with the literature, but obviously that's not going to be the case. Like every year into the future, I hope that there'll be a pathogen that obsesses us in the same way. And so yeah, maybe this is a nice slice of life that we could be having after some of this more pertinent information about COVID kind of abates a little, and we can focus on all the other things because plenty of research has been going on behind the scenes, right?
Right?
We may not have heard about it that much in the media because the media pays attention to what it's going to pay attention to. So yeah, I think it's a really nice idea that we decided to do this.
Yeah, I mean, so what I did was I went through a couple of microbiology, not even microbiology, so scientific repositories, which list how many articles get cited. So a citation is when a piece of research gets cited by another researcher. So it's almost like kind of a metric to check whether someone's read the research and whether it's been important enough for it to be involved in other research.
Yeah, like the likes of the academic community. It's the thumbs up of the academic communities.
Yeah, I mean, it's almost like the Twitter mentions where someone has an opinion and you just mention it because it's relevant to what you're talking about, or whether you want to troll them, depending on which. But it's still usage. It's still so.
Yeah, that's actually a good point. Like, it can also sometimes draw your eyes to certain controversies, right? Like, that's also a driver of citations.
Yes, indeed. Looking through this, my only criteria was to have something that isn't related to COVID. So we've got a really wide variety of different microbiology papers that explore almost like all the different diverse facets of it. So I'm hoping this will give you a nice overview of different parts of things. So without further ado, let's go on to the first one, which is
Nice.
oh wait, hon, this isn't the first one. The first one I wanted to talk about was
Sophist?
syntrophous conductive pili demonstrate that common hydrogen donating syntrophs can have direct electron transfer option. So this is complicated.
No, it's interesting. I actually had heard about electrically conductive pili beforehand, before like hearing about this paper. There was like a big debate whether or not it was even out there in life, right? Like because pili, right, are these structures from bacteria. They're sort of like hairy, long, proteinaceous, hairy things that come off of the surface. People think of them as for attachment, sometimes sticky, maybe motility, moving around. Certainly, they can also protect the surface if they're dense enough, right? They can like act as like a barrier. And people had hypothesized that they could potentially transmit energy through them. And I remember reading some papers maybe a few years ago, and they were talking about, yeah, like how do you even go about proving that that these things were transferring energy? And it sounds like here they have some evidence that uh, stuff actually grows because, like, right, without these pili, you wouldn't be able to get the energy, so they're not growing if they don't have
Yeah.
them.
So this is interesting because so this entire thing relates around like say bacteria that you find that degrade methane. So this is a big like kind of unknown because very so when organic waste degrades, it can produce methane, which historically has been used to generate power. It's like kind of something people are quite interested in, and but the the mechanism that allows methane to reproduce is a bit more complicated than you might think. I mean, the bacteria that need to break down methane, it's not they need to break down any like these weird organic compounds that you find in say sewage and stuff. It takes a lot of
Yeah.
energy, and they're not able to do it alone. So there's this.
And I think often when we think about methane production too, it also happens in a very specific band of like ecology,
Mm,
right? Like you can't just have any old place to make methane. And I think part of that is because of the difficult chemical reactions that you're describing. Like they have to occur at a very specific point, like in the environment, right? Like not too much oxygen, not no oxygen. That's often like where you think of methane production happening.
uh,
Um,
yeah, and in this case, this is in a like anaerobic environment where there's no oxygen, and when you don't have oxygen, that changes chemistry a lot. So you don't because
mhm.
oxygen acts as like almost a way to transfer electrons around, transfer charge and energy different places, and
Mhm.
um, so so in these sorts of regions, there's this kind of form of life kind of style called syntrophy, where so you've got like say there's autotrophy where plants get like energy from the sun. There's heterotrophy where organisms eat each other. Syntrophy is where they kind of share the load of eating stuff. So they're almost like
Mhm.
sharing organic compounds. In this case, they're transferring electrons.
We're heterotrophs, right? We're heterotrophs. Plants are autotrophs.
Yeah.
So syntropy is this kind of strange in between, where I guess like the community is the digestive system. Like it's the members of the community that kind of make up the digestive system.
Yeah, yeah, I think that's so. I think what they found is like there's some methanogenic bacteria that require another bacteria with them that transfers electrons to them in order for them to be able to produce methane. And the thing is, like, there's been so this is basically speaking to that kind of discussion within the field of whether like how that electron transfer happens. So there, so one of the
Yeah, and I guess. Oh, sorry. Go
no,
ahead.
you go for it. Go for it.
I just want to say, and that's really cool because that's, as I was saying, I'd heard about these structures before. It's nice to give them like a function inside of like an ecology and say, like, this is a place that they could be used. Yeah, because otherwise you're just like, I think you had found a paper like this too, but like people have been obsessed about it because like, oh, you can like build tiny like structures, right, that conduct charge. That was why
Yes.
like electroconductive pili, nanowires, I think is how people talk about it. But from the perspective of like microbiologists who are interested in nature, you're kind of like, okay, but why do they even have them?
Yeah. Old school version, they thought that they transferred electrons via like hydrogen, which is just basically producing hydrogen gas that would be broken down into hydrogen ions. And
Mhm.
there's some organisms that can take hydrogen gas and turn it hydrogen ions, and others that can produce it. Another way is
Mhm.
using formate as that. So formic acid as a way of transferring electrons through. And so that's what the so with Syntrophus, that's literally like this is a model organism for that. Um,
Mhm.
and the only time they found like pili that are electroconductive are in this other genus called Geobacter. So
Yes.
the scientists here have been like had this idea that actually these pili that can conduct electricity are much more common in nature than we thought. And in order to demonstrate this, they looked
Mhm.
at Syntrophus and they took it under microscope, looked at the pili, and they do all sorts of like cool stuff. So they do this thing like electroconductive imaging, where they aim to like
Mhm.
image the electrons as they go through the pili to really basically this to prove that they produce pili that are electroconductive and that it helps it with this entire like methane digestion. So
Yeah,
so
yeah, yeah. No, I think that's really cool. I mean, if we choose it, then there'll be tons of those types of images to look at, like these techniques for figuring like, yeah, what is a electroconductive pili? How do you determine whether or not something is using energy from one source or another? I think that I'm not sure if they do that. It doesn't seem like they do.
yeah. It
Um,
mostly seems like they compare it against other species that form electroconductive pili. So Geobacter
right.
sulfurilucens is probably the main model they're using, and this looks pretty. If they can prove that this moves the same way, then they can prove that it isn't a pili. So one of the things they do is actually like cut out the genes for this pili, put it into Geobacter, and see whether it does the same thing in there. Because if it didn't, then it wouldn't
Mm,
work, and we
mm.
wouldn't be able to. So there's
Sure. Yeah. Yeah, so this is kind of broadening our idea. More things could have pili. I guess we still they and it gives us this hypothesis of like where it could be useful in the ecology. But yeah, that is the conversation that this paper is tying into, right?
yeah.
Like where are they used? How are they used in nature? Here's another organism that might have them. How is that going to advance the discussion?
Yeah,
So yeah, that's
it's
pretty cool.
pretty cool. I mean, again, you never get this
Uh,
from medicine when in coronavirus or other medically related bacteria, because in environmental bacteria there's so much cool stuff. Like it's almost like a random like test laboratory for how life should work. In
Totally. Yeah, there's just so many other environments that aren't the human body. I mean, like from the infectious disease standpoint, right? Like in some ways you can think of environmental microbiology as like an obsession with the human body as the environment. But the Earth's a big place. There's a lot of different places you could live. And so adaptations can be really wild in some of those other places. And I think wild in this way is just relative, right? Like wild because I'm always thinking about the human
yeah,
body. Like then like you tell me about like some zone with no oxygen. I'm like, what? I can't live there. Why do I care? Well, stuff does live there, and how it does it is
yeah,
miraculous.
and like this entire idea of syntrophy, which I literally learned about yesterday when I was researching it. So I'm by no means an expert, but it just seems fascinating that we've got two bacteria that are like in such like in depth like kind of cooperation that they're exchanging electrons. I mean, that's really amazing to me.
Yeah.
And they're two different
That's a
species.
yeah. Okay. We're probably talking about this too much, but I just want to throw out there that that's kind of like electron transport chain inside of our
Yes.
cells, right? Is passing off electrons to different proteins in order to make ATP. Like this is that happening, but at the scale between
Yeah.
organisms.
I mean, I was going to make the point later that it's almost like that the relationship between mitochondria and the eukaryotic cells. So we'll probably come back to this point because there are a few other papers that we're going to be talking about that we'll touch on there. Um,
Yeah.
but now,
Busting forward, we have evidence for HIV cure after CCR5 delta 32 allogeneic hematopoietic stem cell transplantation, 30 months post analytical treatment interruption, a case report.
yeah, this is actually something that did kind of almost hit the news a fair bit. Um, because this is about the cure for HIV-1. So,
I think you got to do this.
yeah,
You got to
the
do,
cure
do the
for
cure.
HIV-1. I mean, they do describe it as a cure in the paper. So, but yeah, I think the thing is like permanent remission is what we'd probably want to use. That, um,
I mean, I also just think that like Q just makes it seem like it's out there for people. This is a case report,
yeah,
right? Like it's like a procedure done on one person. Like, is it going to be applicable to everyone? It's like a very specific type of information that you're getting out. But I mean, this touches like I feel like this can be popular for many strange reasons, right? Like this is the deletion that was presumably introduced into those two twins that were CRISPR'd
I mean,
is the delta CR5 mutation.
yeah, so for the background, HIV is a virus that attacks the immune system. It's very, very bad because it can also adapt very quickly to various things. So vaccines and things are very hard to develop against it. I mean, we only just gotten antibiotics that are just good enough for it. Um,
Part of the reason what makes it so difficult is it targets immune cells, T lymphocytes. We've talked about those tons, and one of the receptors that it uses for entry is this CCR5. And it's known that in the human population, there's some amount of people that have this natural mutation, delta CCR5, and they seem to be fine, right? So there's been an ongoing thought inside of the field that, like, if you just had this particular genetic loci, you wouldn't be sensitive to infection.
yeah, and they actually have studies. There are people in the population who are resistant to HIV because of the CCR5 mutation. And in this case, they had a patient who was undergoing therapy for leukemia. And as part of that, leukemia is like the cancer of the white blood cells, which means they had to find a way to get rid of all the white blood cells, which they did via whole body irradiation. and then they replace that so you can get a donor who with normal like blood cells and donate the stem cells and implant them in the bone marrow, and then restart your entire immune system. So they used a donor who was from this resistant population and gave it to the and this paper is essentially them trying to check everywhere to see whether there's any active HIV in this person's body. And
Totally. Yeah. And I guess yeah, it's very cool from that perspective, right? To know, like, this is something that people talk about a lot. You can see it, but this is like the case was they used the treatment in cancer, right? In this circumstance, right? But they end up being able to get information about this genetic modification, essentially, right? It's a type of yeah, I mean, that's what's happening, right? It's as if, right, that their cells were modified to not have this. But that's why I'm like, oh, this is like, to me, that's probably why it hit the
mm,
news, right? And why it's such a hot topic, because it's actually very close to the discussion of, right, should we be genetically modifying ourselves, right, to have resistance to
yeah,
disease, right? And what does the path of that technology look like? This is sort of the first efficacy point that people could be talking about, I would say.
yeah, and I think the thing is that this they've done this before. So there's a Berlin patient and the London patient who, and this is two times where it's worked, and there are times we don't hear about where it hasn't worked. So this is why it's a case study, not a research like kind of like how
Yeah,
clinical trials.
yeah, and that's why I push back on the
Yeah,
cure,
it's
right? Because it's kind of like I think that like that's a common thing in the media, right? Like to say cure, and then that gets people's hopes up in a certain way. But like these are very invasive techniques, right? Whole like these be like cancer, whole body irradiation, stem cell transplant. Like that's not a simple procedure by any means, and also not operationalized within the healthcare system in a way that it's like, okay, we do these on regular. Right? It's not a kidney transplant.
yeah.
Um,
Yeah, no, it's
so
really hard when to tell someone that the first step to getting treatment for HIV is to get leukemia. I mean, I feel like that, but yeah, but I think as a proof of concept, it does help bolster that idea of the only way to really get rid of HIV is to use these kind of drastic gene therapy treatments,
yeah, I mean, yeah,
or
definitely.
just like
It's a very
or like
perspective.
focusing on CCR5 exclusively and drugs that maybe mimic that. Who knows? But I'm not
Yeah.
very experienced with HIV, so I don't know about how. Oh,
And the scientific field itself has been working on HIV for a very, very long
yeah, it
time.
is a very very difficult virus to deal with. So so it's understandable this drastic method, and it's quite a positive thing that there is some hope out there, and it shows that okay, yeah,
Yeah.
there's something good. But yeah, so that's actually some
Okay.
good news that happened in the coronavirus here. So, um,
There's plenty of good
yeah,
news, but even just the vaccine development has been
yeah,
amazing
it's true.
news.
Fair
I don't
enough.
want to
Okay, so next paper, obesity-related microenvironment promotes emergence of virulent influenza strains. Um,
mm.
so
Yeah, this is this was a very interesting one. I mean, I think there have been obesity models, right, around for a long time to study obesity. So like different ways of getting mice
mm,
fat, and then seeing like how that changes their particular body chemistry and how that might interact with a whole host of outcomes. So this is basically this is the cross with influenza.
yeah, so this was an interesting one because reading it, it was I was very unsure of like why this was so successful. Then I looked at the citations, and I'll talk about it later. But what they did here was they took obese mice, and then they gave them influenza, and they found that they got sicker than the metabolically normal mice, and
Yep.
then they
Which is almost no surprise, right? That's not the super surprising thing, but they have a model to study it, so going deeper is going to be where the
yeah,
interest
they did
comes.
some. They passaged it, so they infected one mouse, and then they took the virus from that mouse, infected another mouse, and found that the virus got more virulent. Whereas if they when they tried that with wild type mice, the wild type with it, so with certain strains of influenza don't infect mice very well. So in this case, they're looking at
Yeah.
H3N2, which doesn't infect mice very well at all. And so they thought, well, since
Mm-hmm.
these obese mice are really good at getting this, how about we can generate a mouse adapted strain using this? So they passaged it, and they found that this strain got more virulent. Um, and
Yeah, and I mean, to me, that locates this. Like as I was saying before, there's this obsession with like the body as the site of of microbiological growth and replication. But this is like to me like definitely like one of those papers that highlights that, right? Like we're thinking about our environment, right? Like this is where the virus
Mm.
lives. Like we are the vessels, and so changes in the vessels can mean a different evolutionary outcome, right, for these viruses. And so that suggestion is just uh, I think this discussion, I hope, has been advanced by having this pandemic, where like health is not just like an individual person, but it's also the population, right? It's also the environment. And I think that this type of information is really great for attaching to
Right.
that, right? Because it's essentially saying, like, if we have certain health deficits in our population, it makes us more susceptible to viral evolution.
Yeah, I mean, I can sort of see where they're going with that. I feel like they this is it's a lot for one paper to just to prove that as a whole, but they do try like, but
Oh,
they
yeah, totally. That's me
but
taking their small thread and amplifying
I
it
mean,
through.
they do mention something like that in their paper. And they also talk about like how there's I think been a lot of discussions about how obesity affects the immune system, and because it was weird because obesity can also do some autoimmune stuff like in generating diabetes. It can also have some like less like kind of lower immune stuff. So in this case, they say that there's a delayed interferon response. They confirm it in the obese population. And the main reason I think that this got so much play is because in the coronavirus outbreak, there's obese populations were much more vulnerable to the coronavirus than people. So this is
Yeah.
yeah. So the main reason this has a lot of citations, it's got published in February, just as the coronavirus was starting, and people were starting to see that. And literally every single citation of this paper is about the SARS-CoV-2 outbreak, and people trying to explain why people living with obesity are such a vulnerable population to it. So
Yeah. And actually, I think that that's a really interesting nugget of information when you think about like models, right? Model
yeah,
systems, right? Like this is a model system, totally different virus, totally different organism, right? But the way that they've used it together and to tell like maybe a pattern basically that we're seeing within this model, then scientists can take that pattern, they can apply it to a different scenario, right? Humans and coronavirus. Again, it's not a one-to-one mapping, right? But like that's what scientific literature is about. It's about sort of like making those connections, seeing those patterns, and then following up, right? So I wouldn't be surprised if maybe some of those papers in that citation list like follow up, right, and say like, oh, look at these obese people that have had bad coronavirus outbreaks or bad coronavirus results. They have deficits in the same pathways that we observed, right, or that
right.
were observed in this particular
Yeah, I
infection.
think that so it came out exactly the right time for that, and also yeah, it does help deepen that our understanding of that interplay. Because it's something that like that has been talked about a fair bit about like the differences in say Western populations and why they're getting more hard hit by coronavirus than say other populations. So
Mm.
there's a vast so this kind of fits in that kind of
Sure. Also, remembering with those observations at the global scale, like covariates
yeah
of like living in different conditions, like all that actually matters a
yeah
lot as
so
well.
much like correlation but not necessarily causation. So
Mhm.
it so this is just one this helps to tell one part of that story I feel. Uh,
Yeah.
So yeah,
Okay. Cool.
the next paper is a very interesting one. I thought um, gut bacterial amyloid
Yeah.
promotes alpha-synuclein aggregation and motor impairment in mice. Uh,
Hmm.
so
This is of the line of research where the gut is
yes,
everything. There's all those. Probably people have seen that before on like all their favorite science news like coverage, right? Where there's so many cells in the gut, there's so many genes in there, right? All this expression, and so scientists have been uncovering all these different ways that it impacts other elements of our
mhm.
health. And in this case, a type of a type of pathogenesis that comes from aggregation of proteins, which is seen in things like dementia and Parkinson's. But like this case, it's a different disease, right? Motor impairment.
Yeah. So So amyloids are essentially, as you said, like big aggregates of cells. So the most famous one is beta amyloid, which is involved in Alzheimer's disease, but also there's α-synuclein, which is involved in Parkinson's disease. But we don't necessarily say that it's affected. So all these researchers have kind of looked around and they've noticed that bacteria also produce amyloid-like proteins.
Hmm.
There's certain strains of E. coli that produces this thing called a curly protein. We're spelt curly with a I instead of a Y, where
Yeah.
which E. coli use to build up their biofilms. So it's kind of meant to
Hmm.
aggregate, and there've been some past experiments that show that that amyloid's kind of like when one encounters another, it kind of changes it in a way that forms aggregates. So almost like the way the
Hmm.
proteins work, but not quite. Um,
Yeah.
so the idea would be that perhaps if, say, you've got your bacterial amyloid and it encounters human amyloid, does it force it to form like an aggregate? Um, so
Hmm.
what they did was they got this like strain of mouse that overexpresses human alpha-synuclein. So this is a mouse model that eventually will get like a Parkinson-like disease. And so what they
Hmm.
do is they give it like different concoctions of gut bacteria to see whether that accelerates its degeneration into motor neuron disease. Um, and so this is like a very extreme proof of concept model where they so because if we use it on a real mouse, it might have, I mean, in a normal mouse, it might take ages for such an effect to appear. But this is almost like just trying to
Yeah.
bias everything so they can show that it can happen, which I think is quite an
Yeah.
interesting thing because again, it's talking about the interaction between the gut and kind of the rest of the body, which is
Mhm.
isn't it? And especially like bacteria as well, and kind of how the microbiome interacts. Um,
Yeah, I mean, this is like they're going to use germ-free animals in this particular
yes,
experiment, right? I think that that's a staple of this type of work, right? Where one of the reasons why we hear so much news about how the gut does this and how the gut does this is because there's this really powerful tool that has existed inside of experimental science, where you can birth mice in a totally sterile environment by C-section, um, and they've never encountered a microbe in their lives,
yeah,
right? And that's like a crazy, basically, I mean, to me that's like space travel but inside of a mouse
yeah,
colony, right? Because creating this totally artificial environment, but biology still works there, and you're able to see the effects of all that other stuff on a living organism, other stuff being
yeah. I
microbiota.
mean, I mean, with a lot of them, they don't grow up very well unless you give them some sort of bacteria to live with. I think the main thing they do here is they show how a bacterial monoculture. So they just give these germ-free mice just one bacteria that produces this a lot that can create effects. I think so.
Yeah.
Yeah, it's almost like a much more advanced test tube, except because you can't really get nerves in a test tube, or then this. So yeah, I mean, this is the intersection between microbiology and physiology and behavior. So again, we've got to cope with interpreting mouse behavioral assessments. So with so which is
Yeah, that's
yeah,
interesting.
there's a lot of
Okay.
bits and bobs in this paper.
I sense that as we go through these, like a lot of these papers, they're gateways to these huge fields in microbiology that, right, like we may see papers like these that touch on these same concepts come up, right, as we continue speaking, right, as we continue covering papers, and we'll be like, yeah, like remember back in 2020 when they did this, right? I mean, that is science,
Yeah,
right? It's like it's always a callback to some research that was done before, and seeing these top papers this year, I'm already getting the sense that we're going across like a really wide range of
yeah,
topics.
completely. I mean, we've dealt with like maybe one pathogenesis paper that was really like very much something that would hit news, and then we've hit like various. So we're going to be hitting lots of different parts of microbiology. And I think the next part
Yep.
is going to be even more different from what we've looked at in the past few papers because we're going to be looking
Totally.
at the cultivation.
Cultivation.
So would you like to go?
Oh, yeah, sure. Cultivation and functional characterization of 7 planctomycetes uncover their unique biology. And so, probably should be no surprise to people that I do not know what planctomycetes are. I've never heard of them before this paper, and that's part of the amazing part of like this study is that they are going in and trying to find things that have never been grown before. And because they've never been grown, we've never studied them in any real way.
Yeah, so I mean, I literally had to go into the Wikipedia and go like, oh, planktomycetes, they're an aquatic microbe that they don't really interact with humans much, so you probably wouldn't. So most people probably wouldn't have heard of them. They have a role in aquatic ecosystems, but I
Mm-hmm.
mean, you can say that about any living thing in an aquatic ecosystem.
Yeah. Well, I mean, that's where the information would come from, right? It's kind of like bulk sequencing, sampling some water. Like we know they're there, they're floating around. What are they doing? How do they live? How do they grow? All those questions. Typically, from the microbiology perspective, we have to be able to cultivate organisms to be able to undergo some of those
Yeah,
studies. Um,
and
and yeah, they do that here, and they find some really weird things,
yeah,
like division plane, I think, is one of the things that they
yeah,
report.
they just report because like they're because these planktomycetes they can some so there's different ways that bacteria can replicate. They can bud off slightly. They can do binary fission where it's like kind of equally they burst into equally sized parts. They do all sorts of weird
Yep.
and the interesting thing about this paper is the fact they got to culture these microbes because there are so many microbes out there that we literally have no idea what they look like, what they or how they behave, and because we don't really replicate, we don't really know how to grow them. So the one of the big
Yeah.
problems, I remember, oh god, about 10 years ago when I was working in like say an aquatic microbiology lab briefly, was that
Uh-huh.
all the tests were designed for medical bacteria. Nothing was available for so
Yeah.
trying to study bacteria with like very limited tools and like you can't put these in an incubator because the reason we have incubators is because they're designed to mimic the insides of human body, but um,
37 degrees,
exactly.
right?
Whereas like aquatic ecosystems, they're much different. So these researchers actually took an approach because I mean, isolating environment, these bacteria have a lot of trouble surviving in captivity. So these researchers basically
Mhm.
took the same kind of ethics as what we do for say a normal animal in captivity, where the thing you need to do is to replicate its natural environment as like robustly as possible, and that's what they did here, which means that they have to be very very patient
Yeah.
because some of these bacteria can grow very very slow. They can take a very long time to grow, and before this, there were like 29 genera of planktomycetes they discovered. Now they added 79 to that, and they've done a lot of yeah.
I think that what you said about like taking them out, like trying to replicate the environment as much as possible, it's like for people who take care of exotic
Mm,
pets, for people who are into exotic pets or aquariums, right? That knowledge is actually probably pretty normal.
yeah,
Like people are like, yeah, of course, when you take an animal in, like you're supposed to give it like some water from its environment, or like right, you like acclimatize it to the tank, or you float the fish in the tank for a little bit before you let it out in the water. But I guess because when microbes are really small, we started to think of them as objects a little bit, not recognizing their own lifestyles that they live out there. And there's been a huge tradition in microbiology of growing things with defined medias, right? We want total control over all these different things. And of course, that's an
mm,
amazing tool to be able to grow things. But if we're going to find information about stuff that we can't grow using those methods, we're going to have to branch off and use some of these techniques. And I've been really excited. I love this field actually, right? Like it's just so cool because to me, like all that 16S sequencing that we saw in the
mm,
last week, right? There's huge branches, right? These huge amounts of things. They're all related. They branch off and they live in different environments. We don't have any view on them except that right now. Like that's the primary view that we have on those microbes is just from sequences that we found in some space,
yes,
right? But knowing the environments that they grow in gives us access and being able to then grow them and study them in those environments, even if they're still a little bit artificial because we've moved them out from, I don't know, daily temperature changes. It's still giving us this insight, like it's bridging the gap a little bit from like being so narrow minded that we can only grow things with defined media, right? And then like kind of being a bit flexible and then getting some information because of that flexibility.
yeah. I think the interesting thing about this is the kind of the way it kind of almost like lays a diss track on people who go around and just sample a bunch of microbiome from the environment and then assume that they know everything
Mm,
based on that. So
sure. Yeah.
because like
Yeah, it shows you that one when you dive that layer deeper, there's all this complexity.
yeah, so they describe like behave. So basically, they look at the genome and they look at the way that it behaves, and they find that there's some things that you wouldn't predict it behaving based on the genome because our genome is based on like what we can grow in, say, already in the lab, but we don't know how that relates to
Mhm,
things outside of the lab. So they have things about like certain big genes are missing that, and so you wouldn't expect it to be able to do like say binary fission, but it does. So it must do it by a different way. So
mhm.
they have yeah, they do some. So this is quite interesting because it's both like looking at things like it's very observational, but it's also kind of very interesting in that it's. So yeah,
Yeah, way it links out. I mean, just yeah, I mean, like, I love this type of stuff because, to me, maybe because when I was going through grad school, like, there was so much 16S
mm,
sequencing, and, like, it was so exciting to learn stuff from there. But at the back of everyone's minds, because we were all, like, classically trained microbiologists at some point, there was that question, like, what would happen if you actually observed these organisms, right? And you would learn so many other things. And this is a great way to bring that into the consciousness of other scientists and the public as well. I think that's always an important thing when we think about covering a paper.
yeah, and has got that spirit of discovery that is. I mean, finding something that no one's ever seen before. Like, hey, I mean, you're probably going to see images of bacteria that people haven't seen. Well, they've seen it now because it's been around since 2020. But I mean, that
Yes, yes. Yeah, that's partially also why I think that I get that space vibe from these things, right? Because like that's the joy that comes from a lot of a coverage of like astronomy and stuff. It's like never been seen before. Thousands-year-old light reaches our eyes. This is the same thing for microbes.
yes, definitely. So that's and so moving on to the next part. Next part is also a very different facet. This is more going into biotech and how we use.
Ah, off the curtails of Nobel Prize
Right.
work, right? That was awarded very recently. Continuous evolution of SPS-Cas9 variants compatible with non-G PAMs. So we've spoken a little bit about CRISPR in the past, right? It's this great technology that we have founded in bacteria, some sort of immune system, but repurposed in the lab, it can edit things. And specifically, it requires one of the things people really love about it is it doesn't require too much it doesn't have a lot of
Mm.
requirements. You can edit anywhere, right? Like in terms of getting it. But one of the requirements that it does have is this PAM sequence. So there is one when we're designing the site of where we're going to edit. There is something that we have to do, right, to that guide RNA, and that's make sure that it can recognize this PAM. But off the back of another wonderful technology, directed evolution, we have we have a way now of nudging those Cas9s to not require right that one requirement that we had in designing their guide RNAs. So yeah, that's kind of cool.
Yeah, I mean this is quite cool because they target evolution so it can to expand the amount of like different sequences it can do, and so that's been. I mean, yeah, it's very biotechy. So there's very much interesting processes
Yeah.
for say they have this protein they want to like direct it to recognize specific sequences that don't have any G's in them, and then they just take us through that process and then about how they evaluate it. They do some like mechanistic work on like how it will be useful. So they test out on a sickle cell gene that wouldn't have been able to be targeted previously, and they show how with how much precision they can edit a gene, but like just by looking at base
Yeah.
by base, which is really interesting. Um,
Yeah, this is down the. I mean, this is I feel like emerging biotech, right, is like this huge area. This technology sits at this very interesting intersection where it's saying like we want to have full control over the genomes, so let's remove all the barriers to that, right? And using this technique of directed evolution, they do that. And I think that that's probably what we'll end up speaking about if we end up choosing this paper is what is this directed evolution technique? How do you harness evolution, right, to go down this path? And it recently won a Nobel Prize. and but the technology itself has been around actually for some time, and it's based. I think the one that they use too is based on one of the oldest oldest
Yeah.
molecular biology tools, 13 phage. So it's a virus, a bacterial virus that at the early days of molecular biology, they used it to replicate
Hmm.
DNA. When we didn't know how DNA replicated, they knew they could make DNA in this phage. They could make circular pieces of DNA in this phage, and over time, that phage is being co-opted into this directed evolution technique. I don't know.
Yeah.
It's a great thing. I could probably talk over hours. We
I
should
will note that down because it's interesting.
yeah.
Yeah, because I mean it's yeah there's a lot to talk about with that paper. And so I guess we can move on to the next paper, which is called let's see if I can still flip it over. Uh, Identification of novel mobile colistin resistance gene MCR10. So yeah,
Mm.
so something that we haven't really talked about too much on the show because we've been focusing so much on virus antibiotic resistance. Be afraid.
Yes, yes. This is like when we were going through grad school, I just remember this was huge, right? Like antibiotic resistance. There were so many reports around the world, right, in the Mortality Morbidity Weekly, right? Like it wasn't this emerging virus; it was antibiotic
Yes.
resistance, right? Like there were. We have these great tools, antibiotics. They've helped suppress infection, right, in many different cases. But evolution is happening, and bacteria are finding ways to subvert this tool that we have, and we don't understand antibiotics well enough. I think is one of the issues
Yeah.
as well. Is that like we use them as this like broad tool to suppress growth or clean things up, but really they have a purpose inside of the environment. They're competition molecules, they're signaling molecules. So biology basically has a lot of ways to work around them is all I'm trying to say, I guess, right?
Yeah.
They're not this totally simple thing. Biology has been living with these molecules for many ages, and to no surprise, as we see more of these variants come out, we are learning about biology now through right the ways in which things subvert the the antibiotic resistance or antibiotics.
Yeah, and colistin, the antibiotic they talk, that's like an antibody of last resort. So of the antibiotics
Mhm.
you don't want to see resistance appearing to, it's colistin because theoretically we shouldn't be because it shouldn't be used enough for that resistance to happen. But obviously,
Yeah,
with a
absolutely.
lot of antibiotics, we didn't invent them. We stole them from bacteria and then co-opted them to use against them. But bacteria have been using them for not centuries, but a long time. So they
Longer than centuries, I
yeah,
bet.
like they've used it for at least a few days. But yeah, um, so there are natural resistance, and they discovered like this antibiotic resistance gene that's attached to this plasmid, and so it can transfer over between different species. And they also like once they figured out that this was related to antibiotic resistance, they went back into all like the gene records of because people researchers are generating records of like what bacteria they find in the environment anyway, and they compared them. So you're like, oh, has anyone seen this before? And they found out, okay, it's in 11 different countries already, and we've only just discovered it. So okay, um,
I mean, that's happening with the variants too, right? That's how we've been coming to the understanding of that
yeah,
phylogeny. Um, I guess to no surprise to people, this title, like one of the reasons why I think it's great to say, we should be cautious and wary and frankly afraid, but not too afraid because don't live your life in fear. MCR10, there's a lot of these colistin resistance cassettes that have been found. This is just a report of a novel
right,
one. Not just a report, but this is a report of a novel one. So it adds to that long line of lists where, yeah, there's diversity in the way that things live in this world, and we're just scratching the surface of that and how that intersects our health, right, and the tools that we use to maintain our health in this in the
yeah.
world.
And there is a lot of continuing work on tracking like where antibiotic resistance is arising, and the discovery of a new one means that this new one is now being cited in almost every single paper that's also investigating this. So
Yeah, yeah. That's a reason why it's like, oh, I guess we didn't explain to anybody, but we've been going up in the number of
yes,
citations as we gone down the list. So we're seeing things. We're getting close. This is top
yeah,
three,
top
right? This is
three.
the
So this is the
third.
third most like cited one, which is with 62 citations, and it kind of it speaks to the kind of how active the field is, that some people are investigating this. So it's not like everybody
Yeah.
dropped everything.
Exactly. And I shouldn't get that's a really good I want to bring that point in because, we've been obsessed about talking about COVID-19 because of its impacts on human health, right? And here we have MCR10, right? Like antibiotic resistance, novel antibiotic resistance in the top three non-COVID-19 things out there. It's important to remember, right? Like before we had this pandemic, there were other big things on scientists' minds that we were trying to deal with, and antibiotic resistance is one of them.
Yeah. Uh, so the next paper is isolation of an archaeon at the prokaryote eukaryote interface.
Ooh, we're back to the sort of historical almost, like trying to dive deep into the history of
Yes.
life, right? What happened way back then? This is about the oh gosh, what is it? I can't remember the term now. Symbiosis, endosymbiotic theory,
Yes.
right? We're trying to get a little bit of information here. Um, they find this Archaea, which is a microbe that has more similarities to eukarya than it does to prokarya, or at least it lies somewhere in between when you array it all in a phylogeny. And that means so there's I guess if you look at the tree of life and you try to like trace it all the way back, it would mean that there is some point, right, in our shared last common ancestor, right, that is, right, the Archaea eukaryote
Yeah.
potentially, right, that hybrid that exists out there.
Yeah, it's a big mystery because current Archaea and eukaryotes, we know genetically there's a lot of similarities, a lot of genes are shared between them, but exactly how it came to be that it got associated with all these other prokaryotes, and like how did the mitochondrion
Yeah.
come to be? How did the nucleus come to be? How did all these different microbial? It's still quite a lot of mystery there, and this.
Yeah, it happened a long time ago, at a time of life that were like lots of single cells around.
Yeah.
Presumably, that's how we think of it. And if you think about fossils, you're not really finding that stuff in
No.
fossils. So I'm always just surprised and amazed of the types of insights that can be got from the past just by looking at other organisms that are living, right, relating them to other things, yeah, and this type of stuff.
So yeah, this looks at like a lineage of Archaea that are called the Asgard Archaea, which are the ones that are most related to the to
It's
eukaryotes.
fun.
I mean, this species actually is like they report on this like the species Archaea. They've been growing up for a decade, so the Archaea grow very slowly. These are so we're talking back to the Methanogen, so almost back to the first paper we are talking about, where these are they eat methane. They take a very long time to go, and they also are kind of syntrophic with other bacteria, so they operate as a community. Um,
Hmm.
so in this case, they had to create a bioreactor to enrich marine microbes for 2,000 days before they could actually see something. They saw like a little bit of cloudiness, and that's how they realized that there's something growing in there.
Yeah, so also back to the planctomycetes, right? And just like to grow certain things, it takes these incredibly complex setups. And after growing it, now they can start to understand, I guess, some of the same things, right? Like they were learning in the planctomycetes. Once you can grow something, you can start understanding more of its biochemistry. You can understand what it eats and what it's dependent on. And I guess the whole idea of why this is significant is, again, because here's something that has features, right, that are halfway in between, right? This is like the missing link almost, maybe, that we're finding, right? In some ways, you could conceptualize it like that between the lineage of Archaea and
Yeah,
Eukarya.
I think that's a good way to time. I mean, it's not because I think the researchers have say that it definitely isn't quite what an exact missing link is, but maybe an ancestor or not, let's say, a descendant of that missing link, another pathway that we can inform us what that missing link would look
Yes.
like. Um,
Yes. Oh, I think that's a much better thing because missing link is like it's in the past, but this is living now,
Right.
right? So this is ascendant from there. It would have branched off from there. It's filling in the tree between this space, and it could show us that it could show us some information because again, no fossils from back
Yeah.
then, or the fossils are very difficult to interpret. So like we're sort of interpreting from living ancestors.
Right. And the interesting thing also I picked up on this was is also like in a syntrophic relationship with another bacteria. So again, it was sharing electrons via hydrogen ions and formate, and so that's again
Mm-hmm.
going back to that previous like thing on syntrophy, and also going towards that relationship between our cells and mitochondria.
Mm-hmm. Mm-hmm. Yep. And oh yeah, because for endosymbiotic theory to work, right, to say that like we took in something, right, that would then live inside of us and create things, that essentially is syntrophy every day,
Yes.
right? Like syntrophy just live between organisms, but like we have that within ourselves, right? Like that, or that's the theory at least, right? That we think that that's happened in the past, when we have these stories to help us support that.
Yeah, and it took. So I just keep going back to how it took them 7 years of in vitro enrichment for them to finally isolate this, because part of the thing that makes things quite difficult is that you have to like get a monoculture in order to identify something. Otherwise, you've got another bacteria. But when you're growing something centrophically, they have to like they had to like switch out its partner to isolate with a different centrophic bacteria, and yeah, so it's this is part observational and part very theoretical. So they have this idea that they propose from this of like kind of the archaea and these like, but forming like almost a network around like other bacteria that it's interacting with, and from there eventually
Mm.
developing into what we know as eukaryotic life. So
It's pretty wild. I mean, I'm actually surprised that this is number two on the list because it's so. I
yeah,
mean, it gives me a lot of hope for the science. Like, I love this type of pure thought, like, in terms of like discovery. Like, it's very discovery. It's very driven not by practicalities. It's driven by the desire to know what went on back then, right? And to support our understanding of life itself. Where did life come from as we know it? And like trying to leverage the diversity of microbes in order to get that insight.
yeah. I mean,
Um,
I think it's quite interesting on its own, just looking at how these archaea are forming these really in-depth relationships, and also it's looking at the. I mean, it's going the same thing of like the planctomycetes and the whole like discovery aspect is really cool. Um,
totally.
so,
Yeah.
and I'd say maybe it does add this to this like idea of the entangle, engulf, endogenize model of like how actually that's another thing that I really like about microbiology. This kind of features is that it's not just about talk about like back the infections. It's talking about like life at its most primordial, at like how kind of back
Mm-hmm.
when life was testing
Mm.
out what it should be, and like
Yeah.
and so, because
Yeah.
essentially we are all descendants of microbes, and so this paper is quite
Indeed.
interesting from that perspective because looking at microbiology, but also looking at everything else that comes from it, and looking at how all the
Yeah.
different ways that these bacteria can live together and cooperate and create energy. That's really fascinating to me, and also fascinating how we all came to be. It's life at like kind of its beta stage, where
Yeah, totally. But even not that, like because presumably this stuff is still
yeah,
out there, right? Like we're getting those insights because we're like we've just never looked for the things that could have been descended from those those prototypes at the beginning. Those prototypes may have been successful in the particular environments that they grew in, right? So they're still there. They're still doing
yeah.
it. Like our world supports a lot of different types of life, not just what we've come to understand thinking as humans do, like about our own bodies.
I'm just thinking about the poor scientist who was doing the same thing but gave up after 5 years because their grant ran out.
Oh
I
no,
mean,
yeah.
that's the because this is the sort of thing that doesn't fit into the current model of like the way. So I'm imagining like maybe like some in 40 years someone will find the something that generally looks like amazing, but it just will take a long
Absolutely.
time and a lot of effort. So that's what I really appreciate about this.
Yeah, I mean that's what I'm saying. Like, it makes me so happy to see this paper in the top cited
Yeah,
list because I'm just like, wow,
they
like go these people. Like they were thinking and they were like not hearing any of the haters as they continued down their path of thought and research.
yeah, I think they really earned that second spot. I mean, they got 109 citations in 1 year, which is quite a lot. The average for microbiology is about 3 citations a year, but so I'm really glad that
Yeah.
they.
here.
Yeah. Uh,
Okay, and in our number one spot, we have, well, number one cited
yes.
spot, a deep learning approach to antibiotic discovery.
Yes.
So, maybe no surprise, taking the buzzword deep learning, attaching it to a huge problem that we already described in the number three slot, right? Antibiotic resistance, and pushing them together. We've got number one, essentially a platform, right? Sort of a drug discovery
Yeah.
platform. One could.
Because as far as I understand it, drug companies have like these massive libraries of random molecules that they don't know what they do. There's like can run with billions, and in order to test every single one, it would take quite a long time. So people are always looking for ways to streamline that process, to find ways to pick out molecules that are likely to have an effect. And this is like the newest way to do
Yeah.
that.
Yes. Yeah, I mean that's so right, one of my past research positions was in crystallography and specifically taking crystallography and using it for drug screening. And one of the things that I remember from back then someone was saying was that you can't find good organic chemists anymore to make these libraries because they've been snapped up by all of the
Mm.
drug companies, right? And they're in there and they're making these huge diverse sets, and it's funny to think that they don't actually know what to use them for. They're just resources that they need at some point, right, to find the next drug for X, Y, and Z. So here is a software method that then gets they validate it right inside of an animal model, which is one of the important things, right, that they're doing to show us the worth of this
Yeah.
software. But then yeah, using software now to cycle through it, right? They don't have to do all these biological tests. Well, the hope is that they won't have to do as many biological tests, right? Because they can do this sorting method to narrow that space, and then they can go into the biological tests. So finally, companies can use. I mean, I'm like bittersweet about this, right? Like I'm so happy we have tools to make new antibiotics, but I'm also kind of bittersweet. I'm like, damn, like I know that drug companies are working on deep learning methods to also find cancer drugs and all sorts of other things that maybe not be so important to global health. But okay, side note.
Yeah, I mean, everyone who can do a deep learning method that can make their life easier is doing it now. I mean, it's one of those tools
Yeah.
that we're going to have to adapt to, and I think the interesting
Mhm.
thing here is that because I think the interesting part of this would be to understand how that deep learning model works, because the important thing is the data going in good? What's it doing with that data? And what can it
Mhm.
I mean, in this case, they actually produce like a new an antibiotic with like a new mode of function or with a structure they haven't seen before. It's called halocidin that
Mhm.
they test out. So part of this isn't just like deep learning project, but they find a new antibiotic, which is quite
Yeah.
a cool finding in itself.
Yeah. Yeah. This antibiotic is something that that we wouldn't have even considered before as an antibiotic. But once again, I think just to pull it full circle a little bit with this research is that like, um, when we found the first antibiotics, right, we didn't have any good understanding of how they worked in the ecology
Mm,
of things. What about this object, right? Like pulling it out of a screen like this, I also think that there's sort of this huge gap of knowledge. Like, yes, it is going to be very useful potentially. It could be very potentially useful as a therapeutic, but like, what else is it? Like, I guess it's a totally synthetic
yeah.
molecule, right? So, um, yeah, I don't know. There's an interesting element here. I'm always like wary of some drug discovery in this way because I think it ignores. It's such a new tech, and it just ignores things like does this get broken down in the
Mm.
environment? Right? Like that's not what this deep learning model told us. This does not tell us about like any of those things. And so yeah,
Well,
anyways, I'm excited about it, but also so wary of like mashing this technology into making biologic molecules that we end up using in our
Well, yeah,
biology.
I mean, I think we've seen a couple of papers like this, especially for antivirals, where people are using these techniques to
Mhm.
almost like do a more advanced way of throwing spaghetti against the wall, where seeing what works
Yes.
and testing it against, like, say, we've seen a lot like with spike protein, where they go, okay, let's see whether these molecules will wedge into a specific crease in spike protein. But of course, that depends on our
Hmm.
understanding of spike protein, and with bacteria, it's more complicated because we're looking at all these interactions with its biology. So again, so almost it's like trying to find. I mean, with a lot of these libraries, there's a concern that if you find any way to segment it down, just by pure chance, you'll find a good like
Yep.
candidate. So
Yeah.
there's that kind of
Yes.
need for understanding of because if we get to the point where we can model an entire bacterial's biology, like in a computer,
Yeah.
then yeah, I
Well, actually, now after hearing you say that, I'm buoyed up a little bit, and I am very excited from the idea that it's a totally new mechanism. New mechanisms mean new studying of biology to understand that mechanism, and thus understanding, right, of those
right.
organisms themselves. So, yeah, there's definitely stuff to be very excited about here. I think sometimes I poo-poo on deep learning a lot, and to see that it's number one, I'm just like, oh, deep learning, thank you.
Oh yeah, no, that's understandable. But yeah,
Okay, we gotta choose something for next week
right,
now.
yeah,
So all of these are so very cool, and I would be happy with any of them almost. Maybe I don't want to do obesity-related
yeah,
microenvironments. I'm not feeling that one. I'm not feeling gut bacterial amyloid. Um, I feel like there will be other microbiome. Those microbiome. One of them's a microbiome study, and the other one is an obesity. Right,
yeah,
essentially the tools of obesity. We will see them again for sure.
yeah.
um,
And yeah, I'm relieved because the amyloid one has a lot about studying mouse behavior, and there's a lot of variance in there that makes me kind of uncomfortable to draw big conclusions about. Um,
yeah,
so
that group the group that published it. I mean, this is again just like from the surface level analysis of this whole paper. Like eLife, I love that journal. That group, I've heard lots of good things about them, but I also know that they are always pushing the envelope in terms of what they can show, and like I don't know how that sort of filters out
right,
actually with the test of time. Yeah.
okay. So I think that can. Um,
Don't want to do HIV
no,
cure, although someday it would be really interesting to talk about it because I actually do think, as I alluded to in this conversation, it is the stepping stone to speak about genetic modification of humans for therapies. Um,
yeah. That leaves us with the planctomycetes paper is quite interesting. There's quite a lot to unpack with that. I feel. Um,
yeah, we get to see EM pictures of microbes never before
yeah,
seen.
you get to have like genomes where they look at kind of different functionality within them. So it's a lot. There's a lot of stuff that we can talk about with that. Um,
Hmm.
there's yeah. What other things are? I mean, there's obviously the top one. We could always talk about deep learning approach to antibiotic discovery, but uh,
We can talk about learning. I'm not against it. It is very interesting. I don't know if we'll get as much insight into the algorithm. I mean, I'm remembering the episode that we did with what was it? Did you? It wasn't Primal. Rosetta. You looked at the
yes.
Rosetta algorithm a little bit. I think that was definitely an easier one than this one. This is gonna be like a black hole potentially, but also, I mean, I'm curious about that sort of stuff, deep learning. I do want to know how it's done and how it
I mean, yeah,
applies.
given that it's like been cited so much, and it's, I do feel like maybe that's the big important paper that we should probably
Yeah.
take a look at.
It, I am happy that it is talking about antibiotic resistance. That's a good reason to cover it as well, because as we've said maybe three times now in this, three distinct times, like that is a really big problem in human health that we should be thinking about. And also, like I think this is gonna sound strange maybe, but like I also think there's like also like there are blinders that we had on when we made our original
Mm.
antibiotics, and so like I would like to know whether this technology is addressing those blinders or whether we're just repeating. We're just gonna go down the path where we're like, oh yeah, we just go out a new antibiotic and we're gonna get resistance a few years down the road again.
Yeah,
um,
I think that I think that's the paper because I mean, it's got lots of citations. It's sort of, people obviously consider it to be important, and I want to see what the fuss is about. What is about this deep learning?
Yeah. Okay, let's do it. I'm happy with that. Again, I think this list we may pull on this list again in coming episodes because there's stuff that I almost don't want. Like I do want to talk about continuous evolution at some point, but I also think there will be more papers in the future that talk about that. But that one particularly, just like to know that people are trying to remove the PAM specificity from Cas. I don't know. That's like you want to edit everything. That's really the strategy. I maybe I'm a bit critical of it. Maybe I don't want to cover it now. But I'm
Yeah,
talking about it
I
out
got the
loud.
impression from that paper
Uh,
was like creating lots and lots of different versions of the cast to suit your functions, and we can sell them to you at a reasonable price. So
but also that you can change anything in your genome. But like, why do we want to do that? Like have a lot of questions here, right? Like
What have you got against like changing our genome? Don't you want to grow wings? Come on.
I think we're not. Yeah, we just haven't spoken about it
Yeah.
enough, like as a
Personally, I
society.
would like an ear
So,
on my back. I think that would be a useful thing for me to have.
I mean, if they could give me a sense organ, I'm all for a new sense organ.
Yeah, I
Like,
could definitely do with a common sense organ. Just
I can tell in the weather. I'm like, oh god, I have a headache. It means that it means a storm's coming. Okay, let's do. So join us next week where we're going to be covering a deep learning approach to antibiotic discovery.
yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get anything right, especially today where we're covering such a broad scope of microbiology that it's likely that our 1 hour look at Wikipedia probably didn't capture everything. So if you have any comments and questions, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is a process, and we're doing it right now. We can all participate, and so we hope that you had a good time listening to us ramble on about microbiology. If you have anything to add, please, or found something unclear, let us know.
It's been a pleasure chatting with you, Danny.
Same here, Faz.
And tune in for more microbiology content next week.